This is the start of our discussion about eukaryotic DNA replication, where we will look into the process of initiation today. The concepts will mainly have a metazoan focus, which means the model systems like humans, mice, and frogs. These concepts are, of course, derived from the knowledge from yeast, because most everything is conserved, except how the origin of replication is defined. Yeast origin, called the ARS as we saw in the origin of replication video, is very, very different. Since initiation as a concept can be very overwhelming and difficult to absorb, I will split the initiation step into 3 parts. These steps are origin recognition, origin licensing, and origin firing. This is the first video, and in this video we will see how the origin is recognized. By the way, the entire initiation can be summed up pretty easily by these steps, which is that origin recognition and definition is done by proteins like ORCs and cdc6. Then licensing is done by the addition of CDT1 and MCM proteins. Finally, origins are fired when CMG helicase and a bunch of other proteins join. Essentially, the 3 videos will expand on these 3 points and hopefully help you understand the process much better. Alright, let's start with the recognition part. So, DNA replication, as we've already discussed, is inherently linked to the cell cycle. In metazoan eukaryotes, especially in somatic cells, this is usually split into interphase and mitosis. The interphase is further divided into G1, S, and G2 phase. And if you know the basics, the S phase is typically when the DNA replication actually occurs. For all intensive purposes, the process of replication per se is technically the process after origins have fired, whereas the G1 phase is used for origin recognition and licensing. This makes sure that all things are ready to start replication in the S phase. The recognition of origin can actually start as early as late telophase during mitosis. The recognition is done by 2 major proteins called ORCs and cdc6. ORCs are the first to bind the putative origin DNA. This ORC stands for Origin Recognition Complex. However, the functionality of origin to move to the next steps occurs only when the cdc6 joins the ORCs. Before we move to recognition, let me quickly review the features of origin of replication in eukaryotes, and specifically the metazoan eukaryotes, that is, everything non-yeast. Simply put, there is no universal signature for origin of replication in metazoans. No motifs, no nothing. But there are some features that are common among the origins. And these are nucleosome-free regions, which mark open and accessible chromatin. We also saw them in transcription. Therefore, logically, many of the origins actually overlap transcriptional start site, or promoter regions. Origins are also enriched for DNA secondary structures like G quadruplexes. They tend to have low complexity regions like CpG islands, which are quite GC rich. In terms of chromatin marks, H4K20 mono or dimethylation seems to be very heavily used to recruit ORCs, since ORCs can recognize some form of methylated histones. And more recent studies have shown a correlation between cohesin loading and collisions hotspots that form these favorable DNA topologies for ORC binding. All of this is in contrast to yeast origin, which has a universal ARS motif structure. Again, watch the origin of replication video for more details on that. One strong feature in eukaryotes is that their linear chromosome structure is actually very large, and this dictates multiple origin of replication requirement. And since there is no universal feature, essentially any potential region of DNA can be used to initiate DNA replication. I say potential because it still needs to fit the common feature requirement. All this jargon is to say that there is little reproducibility in the way origins across chromosomes are recognized and used. Let me illustrate. Say this piece of very long DNA has certain regions recognized by ORCs as origin of replication. But when it comes to usage, most of these are never used. So when you look at replication bubbles, you will find, say, these two origins being used. Also, if we actually quantify, we know that there are about 40,000 to 100,000 origins that are recognized and licensed per DNA replication initiation cycle. And out of these, only a small 10 to 20% are actually used for replication in the S phase. The point still remains that eukaryotes have multiple replicons at any given instance during DNA replication. Now let's get into the ORC and cdc6 and understand how origin binding and recognition is done, primarily by ORCs, and then later cdc6 joins the ORCs. Since all of this is connected to the cell cycle, let me give you some intuitive idea on how this plays out during the cell cycle. In this plot, we will look at protein levels of ORCs and cdc6 in the telophase, G1 phase, and the S phase. That means the cell exits from mitosis and enters the interphase. If we talk about ORCs, it starts expressing in the late telophase and peaks in the G1 phase, and it is not seen in the S phase. The cdc6 expression is limited to the G1 phase, and it sharply drops in levels in the early S phase. Since ORCs start expressing early, the origin binding can actually start as early as the late telophase of the cell cycle. By the way, cdc6 is also called cell division cycle 6 protein. Alright, given this head start and how it plays out with the ORCs are the first ones to arrive and recognize the DNA. A few details on the ORCs: since it is a complex, it is made of 6 proteins, named ORC1, 2, 3, 4, 5, and 6. Their names are ordered to reflect the size of the protein. So ORC1 is the largest, and the ORC6 is the smallest. Most of the ORCs have ATPase domains and winged helix domains for DNA binding. They also have helix loops and helix-turn-helix structures for DNA binding. cdc6, as we will see later, binds to these ATPase domains of ORC1 specifically. And something to note is that ORC6 has a TF2B-like domain. I hope you remember TF2B from the promoter recognition in eukaryotic transcription videos. This is interesting because we said in the features of the origin that a lot of them happen to be around transcription start sites, and ORC6 might have something to do with that. Visually speaking, this ORC looks like a crescent moon or half-ring complex. Okay, so origin recognition is performed by the binding of ORCs, and then finally these origins are converted to functional or initiation origins. Let's discuss how recognition might occur. I will cover some possible examples to illustrate this, and these are not exclusive events, which means that multiple of these events can happen at the same time. The first is the nucleosome-free regions, promoter regions, or CPG islands. This one is a little straightforward because any DNA binding domain in the ORCs like winged helix, ATPase domain, Walker motif, and even the TF2B domain in the ORC6 can recognize DNA that is available for binding in the open conformation, meaning that it is not heavily associated with histones. And this type of feature is predominantly found near promoters. The second example is the recognition of HP1 protein, which is a heterochromatin protein. And these regions are opposite of the regions in example 1. Because in this case, the DNA is not accessible because it is tightly compacted by histones. And it may have heavy repressive methylation marks. But some ORC-associated proteins like LRWD1 or ORCs can attract ORCs to these sites to initiate the process of origin recognition. Third example is of histone modification, like H4K20 mono or dimethylation, which is recognized by ORC1 specifically and its N-terminal domain, which further has a BAH domain. Essentially, histone tails with this modification can attract ORC1 to this region, and that can lead to defining the origin sequence. The fourth example is less commonly observed. It is at telomeres, which has many proteins like the sheltrin complex associated with it. This sheltrin complex has a protein called TRF2. which can attract ORCs to the telomeres to start replication from the ends of the chromosome. So just to reiterate, these examples are not exclusive events. So multiple of these events can be happening at the same time at the same spot. So this is how recognition is set up. Now let's check out the binding dynamics of ORCs. Typically, in the free-floating ORC complex, the ORC1 and ORC4 inhibit each other's activity. Through some regulation that is linked to cell cycle transitions and the ATPase activity of the ORC1 is somehow activated, and that releases this autoinhibition. And only then is the ORC complex able to bind the DNA. And if the ORC assembly is mediated by the ORC4, then it is likely to be on the AT-rich regions. Also, within the complex, there is an assembly order of proteins, which means that certain ORC proteins are next to each other in this ring shape. You don't have to remember this, but here is the order. Notice that these two autoinhibitory proteins are next to each other in the assembly order. I just wanted to give you an insight into the fact that these things happen for a certain reason. Alright, now through some recognition mechanism that we discussed, the ORCs will assemble on the DNA. The complex takes a ring shape when it binds, and this binding changes the ORC conformation in such a way that it is now able to bend the DNA slightly. This bend and non-bend form can coexist, which means it is only a transient state. This all happens in the late telophase or early G1 phase, until cdc6 is expressed in the G1 phase. The conformational change in the ORC also exposes the AAA plus domains of the ORC1, and this allows cdc6 to bind to it, when it is available, of course, in the G1 phase. And now we get a more stable complex of ORC and cdc6 at the origin, where cdc6 and ORC1 specifically positively regulate each other to maintain physical interaction. This mutual binding is what defines the origin as the functional origin. This simply means that this particular origin containing ORC and cdc6 can be used for the next step, which is origin licensing. This functional origin definition peaks in the G1 phase when the ORCs and the cdc6 are expressed at the highest level. Now, as the G1 phase continues, somewhere in the late G1 phase, a new protein complex called CDK1, which is a checkpoint regulator for G1 and S phase transition, starts being expressed. And CDK1 inhibits cdc6 by phosphorylating it. And you cannot make functional origins if cdc6 is not available. And this is the reason why functional origins can only be defined in the G1 phase and not beyond that. In the next video, we will see how functional origins are licensed for replication.